Hello again. Welcome to another exciting installment of lipids and carbohydrates. Last lecture I focused mainly on carbohydrates. Today's this lecture is going to be focused more on lipids, the structures of lipids and what roles they play. Again, at a very high level, just to introduce you guys to carbohydrates and lipids for later lectures. This lecture I'm going to give a brief introduction of lipid functions, talk about lipid characteristics and categories, talk about fatty acids, glycerides or acylglycerols, phospholipids, sphingolipids, and also talk a little bit about sterols and hormones, as well as terpenes and a couple other lipids. We talked about last time lipid functions. One of the main lipid functions is energy storage. Triglycerides or fats are degraded into glycerol and free fatty acids in response to hormone signals. And again, when we talk about lipid metabolism, we'll talk a little bit more about that. But they can be released in the plasma for metabolism in the primary muscle and liver. Lipids can also be turned into soaps by hydrolysis by a strong base such as sodium hydroxide. One of the products, the sodium salt of the fatty acid, is the actual soap itself. So when we think about detergents, soaps, these are actually fats that have been hydrolyzed. Lipids are also important structural elements of cells and organelles. They're found in vesicles, liposomes, membranes, plasma membranes, and aqueous environments. We'll talk about how those structures work. They're also important in signal transduction, so they can be precursors to hormones, again, which we'll talk about. Showed this slide last time, but carbohydrates and lipids, again, are involved in a lot of different pathways, and you'll talk about these pathways in the coming weeks. Let's talk about lipid characteristics and their categories. Lipids in general are small, hydrophobic, or amphiphilic molecules. So amphiphilic meaning they have both a hydrophobic and a hydrophilic component. They form structures such as vesicles, liposomes, micelles, or membranes in aqueous environments. Some of these structures are shown here, where the polar group is denoted by the little circles, and the fatty group or hydrophobic group is shown by the straight lines here. So a vesicle in an aqueous environment is shown here, so the polar groups would be facing the water on the inside and the outside of the vesicle, and the hydrophobic components essentially hide from water on the inside. You can also form micelles, so just a single layer. You can form an inverted micelle, and if you have example in a non-polar solvent, you'd form an inverted micelle. And then you can also form a lipid bilayer. And lipids are extractable by nonpolar solvents such as chloroform and methanol. Lipids can include fats, waxes, sterols, monodiglycerides, phospholipids, etc. We'll talk about those. But they originate from one of two building blocks, from ketoacyl or isoprene groups. Fatty acids are the simplest form of lipids, and they're primarily found in plasma. So when we think about a triglyceride structure, you have the glycerol group and then three fatty acids around that glycerol group. These triglycerides are the solid storage form of lipids, and they're primarily found in adipose tissue. Phospholipids are very similar to triglycerides, except for you have 1 to 2 fatty acids. In the case of lysophospholipids, you just have one fatty acid. And they also have a phosphate group, and they can be decorated with some sort of head group. And we'll talk about some of those examples in a minute. Lipids can also be sterol containing, such as cholesterol, and compounds such as glycosphingolipids. They can be linked with biological membranes. Let's dig in a little bit more on fatty acids. Fatty acids are just carboxylic acids with a long aliphatic chain. So they usually have an even number of carbons. They're usually unbranched, and they can be saturated or unsaturated. And if they're unsaturated, they're usually in the cis conformation, and we'll talk about that. Some examples here are palmitic acid. Sometimes you'll see it written as 16O, which means it has 16 carbons and 0 double bonds. Stearic acid, 18 carbons, 0 double bonds. Oleic acid has 18 carbons and 1 double bond. They are numbered from the carboxyl end. So C 1, 2, 3, all the way down to 18. Some common fatty acids are the short chain fatty acids. These can be 2 to 4 carbon atoms, such as butyric acid, which is C4. They can be medium chain, 6 to 10 carbon atoms, and they could be long chain, 12 to 26 carbon atoms. Again, the most notable one is palmitic acid, is C16. They can be essential and non-essential, just like amino acids. So some that are essential, they cannot be biosynthesized by humans, and then the non-essential ones, they can be made by humans. Two important examples of essential fatty acids that we cannot make, but that our body needs, are linoleic and linolenic. So linoleic and linolenic are both 18 carbon fatty acids, although linoleic has 2 double bonds and linolenic has 3 double bonds. So double bonds meaning they are unsaturated fatty acids. So if you have a mono unsaturated fatty acid, you have a single double bond. Again, it's in a cis conformation. An example there would be oleic acid, as I mentioned. You can also have polyunsaturated. You can have 2 or more double bonds. These are not back to back, so the double bonds are not back to back. They'd be separated by a methylene group. For example, linoleic acid is shown here. Actually, yeah, linoleic acid is shown in this diagram here. The polyunsaturated are classified into 2 groups: omega-3, where the first double bonds appears 3 carbons from the terminal methyl group. So this would be an omega-3 double bond, and the numbering of fatty acids starts from the carboxyl group, but when you talk about polyunsaturated fatty acids, omega-3, the numbering refers to the number of carbons away from the terminal methyl group, and then omega-6 would be the first double bond appears 6 carbons away from the methyl group. Let's talk a little bit about the physical properties of fatty acids. So they're largely determined by the length and the degree of unsaturation of the hydrocarbon chain. So it's basically how long is the hydrophobic tail and how many elements of unsaturation do you have, or how many double bonds do you have. So the relationships are pretty simple. The longer the fatty acid chain length, the poorer solubility in water. So this makes sense, right? You have more hydrophobicity, more carbons, and you get poor solubility in water. So the carboxylic acid end is polar, so you get some water solubility there, but the longer carbon chain you have, the poor solubility, and also the longer chain you have, the higher the melting point. Now, when you talk about double bonds, the fewer double bonds that you have in the fatty acid, the lower the solubility in the water. So the more double bonds you have, you actually get a little bit higher solubility in water. And the melting point decreases with the number of double bonds. This is because cis double bonds place a kink in the linear structure, as you can see by this schematic here. And so they can't get close packing, and therefore it requires a lower temperature for freezing. So this is why you have a lower melting point for things like oils. They have elements of unsaturation in them, as opposed to things like butter and waxes that have a higher melting point. Let's talk through a couple of examples here of saturated fatty acids versus unsaturated fatty acids. and focus mainly on the melting point. So as you can see, the saturated fatty acids, as you go from 12 to 14, 16, larger and larger number of carbons on the chain, your melting point goes up. So these at room temperature, these are in a solid state. So room temperature, around 25 degrees C. Unsaturated fatty acids, 16.1, you can see that the melting point is negative 0.5 degrees C compared to 16.0, which is 63 degrees C. So just introducing one double bond dramatically reduces the melting point. And so this would be a liquid at room temperature. But the same relationship holds true as you add more and more, carbons, the melting point goes up. As you add more elements of unsaturation, the melting point drops back down again. Waxes are esters of long-chain saturated and unsaturated fatty acids with long-chain alcohols. So these have very high melting points. They're useful for a lot of pharmaceuticals, so you'll find waxes in creams and ointments. Carnauba wax is an example. So this is a hard wax that's used on cars and boats. Our skin glands actually secrete waxes to protect hair and skin to keep it lubricated and waterproof. Oils can be mixtures that contain triglycerides, fatty acids, and other compounds. Their melting points usually aren't sharp because they have these mixtures of different compounds. They obviously don't mix with water, so they form emulsions. And pure fats and oils are usually colorless, are colorless and odorless. The color and odor is a result of actually of contaminants that are in the oils. So, for example, olive, corn, and peanut oil, they have different odors and colors due to the contaminants that are contained in those oils. Let's talk a little bit more about fatty acid double bonds and the conformation there. So most fatty acid double bonds are found in the cis conformation, so it's shown here. The cis means the adjacent hydrogen atoms are on the same side of the double bond. So it's not drawn here, but you can imagine a hydrogen here and here. Whereas trans, the adjacent hydrogen atoms are on the opposite side of the double bonds. So for example, you'd have a hydrogen here and a hydrogen here for the cis conformation of this fatty acid. So you can see the trans bonds fix fatty acids in this extended conformation. So they pack similarly to saturated fatty acids and they have much higher melting points. Whereas cis bonds fix fatty acids in this kinked conformation. Trans fatty acids are often produced from unsaturated fatty acids by hydrogenation. So when you hear about trans fats, this is what they're talking about. And it raises the melting points of oils. And the oxidation of these double bonds, so this can get oxidized easier, it can result in cleavage to aldehydes and carboxylic acid, which can lead to oils becoming rancid. Fatty acids in the body, so they're predominantly formed in the liver, and we'll talk about how these fatty acids are made in the lipid metabolism lectures. They're predominantly formed in the liver as well as the adipose tissue, and during lactation they're also formed in the mammary glands. Unsaturated fatty acids account for about 2/3 of all fatty acids in the body. Notable examples again are oleate, which accounts for about 1/2 of the total fatty acids in our body, and palmitate account for about 1/4 of the total. Let's talk a little bit about glycerides or glycerols as we build up to triglycerides. So these fatty acids are rarely found just floating around in solution. They're usually complex to serum albumin or they exist as acylglycerols. So we have a glycerol backbone, and if you attach a fatty acid to it, it's known as an acylglycerol. So these can be monoglycerides, diglycerides, or triglycerides. If we have the carboxylate, the salt or the ester, you no longer bear a negative charge, and these become very nonpolar and insoluble in water. The advantage of using triglycerides as a storage fuel rather than polysaccharides is because more energy is present. The fatty acids are more reduced than the carbohydrates. And again, we'll talk a little bit more about this in coming lectures. But fatty acids are nonpolar and they're non-hydrated by water. And so the triglycerides have more calorie value because they are more reduced than carbohydrates. To break down triacylglycerols, we have things called lipases that hydrolyze the ester bonds to form glycerol and fatty acids for fuel as well as for other purposes. And again, you can also form soaps by breaking these down with things like with bases such as sodium hydroxide. That's triacylglycerols. Again, phospholipids are pretty similar in that they have the glycerol backbone and then fatty acids, but they also have a phosphate and a polar, they can have a polar head group attached to them. So this SN3 position on glycerol is occupied by a phosphate and usually an amino compound. So it can be something like choline, which has a positive charge, serine, ethanolamine, as well as inositol. This inositol, phosphatidylinositol, is an important one that's involved in cell signaling. Platelet activating factor is an important phospholipid. It's similar to phosphatidylcholine, but it contains a methyl group instead of the acyl chain. So at the 2 position, it has a methyl group instead of the long acyl chain here. It's a major mediator of inflammation, platelet aggregation, and membrane permeability. Let's introduce sphingolipids. Sphingolipids are very similar to phospholipids, but the backbone itself is not glycerol. So the backbone is actually sphingosine, which is a long chain amino alcohol. And the fatty acid is joined to the sphingosine via an amide linkage, shown here, rather than the ester linkage that you have with glycerol. But similar to phospholipids and sphingolipids, you can have various head groups, including sugars. So you can have, if the head group, for example, is just a hydrogen, this would be ceramide. Sphingomyelin, the head group would be phosphocholine. You can see some other examples here. Sphingolipids are found largely in the outer face of plasma membrane, and these are important because they're involved in cell surface recognition, for example, in red blood cells. And we'll talk about that shortly as well. So red blood cells, as I mentioned, the blood groups are actually determined in part by the type of sugars that are located on the head group. So if you have an N-acetyl galactosamine, you'd be considered the A blood type, and it's a little bit more complex than this, but essentially these are the main determination of your blood group. So if you have N-acetyl galactosamine, this is considered the A antigen, which would be the A blood type. If you are the B blood type, you have galactose group there. If you have a mixture of these two, both the A and B, you'd be AB type. And if you have none of them, you're the O antigen or the O type. And so this is why O's would be considered the universal donor, because you don't have either the A or the B antigen. And so you can give blood to any type, whereas others you have to have the same antigen type. Let's switch gears a little bit to talk about sterols and the hormones, the other kind of main class of lipids. Sterols are structural lipids that are present in the membranes of most eukaryotic cells. So, for example, in plants, fungi, animals, are you'll have cholesterol, but plants and fungi have very similar structures to cholesterol that are found in the membranes. You have, in all these cases, you have 4 fused rings, and you have 3 6-membered rings and 1 5-member ring. And it's in this planar conformation that gives structure and rigidity to membranes. As you can see, these are very hydrophobic, so they're lipid soluble, and they can diffuse freely through cell membranes into the cytoplasm of target cells. Cholesterol is only found in animal fats. It's primarily synthesized in the liver, and we'll talk at a very high level of what that biosynthetic pathway looks like. The degradation also occurs in the liver. Again, it serves as a component of membranes of cells, and it's a precursor to steroid hormones and bile acids, again, which we'll talk about in future lectures. Here's some examples of steroid hormones. As you can see, they have very similar backbones to cholesterol, except for they are decorated with different hydroxyl groups and methyl groups. And they don't have this alkyl chain that's hanging off the cholesterol group. So you can see these are a little bit more polar than cholesterol, and they can move through the bloodstream by attaching to protein carriers. There are 5 different groups based on their binding receptors, and you can see those groups listed here. But the important thing to understand is these things are derived from cholesterol, and they are more polar due to these decorations and not having the acyl chain. The cholesterol is built up from a terpene or isoprene unit. So let's talk about how we can move all the way up to cholesterol from an isoprene unit. The isoprene, the terpenes are simple lipids that lack the fatty acid component. So they are combined by forming 2 or more of these isoprene units shown here. So if you have a monoterpene, this is 2 isoprene units, and then you can move up from there. Monoterpenes are important because these are involved in flavors and odors, so lemons and citronella, these are all terpenes. These are all monoterpenes. As you can see, there's a couple of isoprene units there, although they may have some additional decorations beyond the isoprene unit. Triterpenes, you can see here squalane, which is an intermediate of cholesterol pathway. You can see how these can start to build up to look like cholesterol. So these are, again, precursors of cholesterol and other steroids. Things can get pretty complex. Tetraterpenes, which have 40 carbon units, these are things like carotenoids, beta carotene, which is a precursor of vitamin A, lycopene, which is found in tomatoes. These are very colorful compounds. You can also have polyisoprenoids or polyprenols. These are side chains of vitamin K, vitamin E, and coenzyme Q. So these are all built from these isoprene unit and to make terpenes, and then very complex terpenes. Just to give an example, some other lipids that are going to show up throughout the lectures here. Last time I talked briefly about LPS, more from the polysaccharide portion of it, but LPS again has this lipid A backbone, which is the hydrophobic anchor, which anchors the LPS molecule into the cell wall of gram-negative bacteria. This thing is what interacts with host TLR4 to initiate this inflammatory response. Again, this can lead to fever, hypertension, and gram-negative sepsis. Some other important lipids, the prostaglandins and eicosanoids, these are local hormones and key mediators of inflammation. They can lead to blood vessel widening or contracting. They can also be used as treatments to treat infants, for example, with congenital heart defects. Leukotrienes is another example of some other important lipids that you'll see. Leukotrienes are made in neutrophils, monocytes, other white blood cells. They are found in the lungs, spleen, brain, and heart, and they are responsible for inflammation and asthma and bronchitis and anaphylaxis. And those structures are shown here. Again, I have the structures up here to give you a flavor of what these things look like, but you're not expected to memorize or be able to draw out the structures. But it's more about understanding sort of the basic components of these things. Lipoproteins, I'm going to introduce this as well. This becomes very important when we talk about lipid metabolism in a couple weeks. Lipoproteins are phospholipid and cholesterol vesicles, as shown by this diagram here. So there's phospholipids on the outside. There can be cholesterol esters on the inside, cholesterol within the membrane. These are important because they are found in the plasma and they transport lipids such as cholesterols and triglycerides throughout the body. And we'll talk about the different types of lipoproteins when we talk about lipid metabolism, but what the lipoprotein has in it helps determine what the lipoprotein's fate is throughout the body. And again, we'll talk about this in a couple weeks in a little bit more detail. You also see lipid-linked proteins throughout biochemistry. These are different from lipoproteins. These are proteins with covalently attached lipids. And so they're typically peripheral membrane proteins, and they have a lipid attached to them. The three common types you'll see are prenylated proteins, proteins that have fatty acids attached to them, and then GPI-linked proteins. So they have glycophosphatidylainositol linked to them. All these things, lipids and proteins and carbohydrates, integrate to form biological membranes. And just to give you guys a flavor that these, we show these things as static things, but in reality, these biological membranes are very fluid. So cells can interact with each other, molecules can interact with each other, and these lipids and carbohydrates are one component of these membranes, as well as floating inside and outside the cell as well. And so hopefully this set you guys up for carbohydrate metabolism, which I believe will be coming up next, and then I'll be coming back to you to talk about lipid metabolism. This last slide I'm just going to leave you with is the study guide. I'm not going to go through it here, but it hopefully helps focus these last two lectures that I gave on lipids and carbohydrates. So thank you for your attention, and I will see you next time.